Hello everybody, today we'll talk about tissue-specific expression of proteins and messenger RNAs. This is lecture 1 from the course Molecular Genetics 1 MLGN 301 2006. So in this lecture we will talk about the tissue-specific expression of proteins and RNA, and there is a recommended reference book for further reading which called Gene Expression 5th edition by David Lec1. The learning objective in this lecture will be learning different methods to study tissue-specific expression of proteins and messenger RNA. The learning outcome of this lecture by the end of this session and practical students are expected to be able to understand tissue-specific expression of proteins, understanding tissue-specific expression of proteins, and of course understanding specific methods for studying the protein composition of tissues, and understanding general methods for studying the protein composition of tissues. Also, by the end of this lecture, the student should be able to understand tissue-specific expression of messenger RNAs, and of course understanding specific methods for studying the mRNA expression in different tissues, understanding general methods for studying the mRNAs expressed in different tissues. We have to understand that the various parts of mammalian body on display differ dramatically in appearance, ranging from muscular legs and hind quarters to the soft tissues of the kidneys and liver. However, all these diverse types of tissues arose from a single cell. This fertilized egg or zygote, raising the question of how this diversity is achieved. So take a small look at the different organs of any human or any organism, and you can see that different organs have different expression level of proteins and of course RNA. So that's why we have to have tools, molecular tools, to study this diversity in different tissues in the same organism. Of course, we have to know that the central dogma in all organisms is genes transcribed into RNA, and RNA and messenger RNAs translated into a protein. This is the central dogma of genetics in all organisms. But in proteins we have another central dogma. Let's talk about central dogma of protein. So when you talk about proteins known for sequences. The sequence of proteins lead to a structure, and the structure leads to a function. So the central dogma of protein and tissue-specific expression of protein are sequence, structure, function. And cell express different subset of the genes in different tissues and under, of course, different conditions. Hence, if the differences in the appearance of mammalian tissues described above are indeed caused by differences in gene expression in different tissues, they should be produced by differences in protein present in these tissues. This means that if you take a look in different organisms, for example, the heart, the brain, the kidney, the liver, any other parts of the body, you will find that there is a different expression, there is different RNA levels and types in different organs. So that's why you will find each one have different proteins, that this protein that make the final structure of the this organ. So different parts of the body have different level and type of expression of RNA, and of course have different type and expression for proteins. And of course the same thing if you want to compare between two different organisms like human and monkey, if you will find also that kind of differences. So there is differences can be detected in both specific method. There is specific method which study the expression of one particular protein, or we have also general method aim to study the expression of all the proteins in a given tissue. So we can compare to find if this specific protein, for example, expressed in different tissues, or which tissue this specific protein expressed in, and this type of method called specific method. And we have other methods called general method, which means that we studying all proteins that are expressed in that type of tissue. For example, we can see all the proteins that expressed in the kidney, in the liver, in the brain. This is called general methods. So in this type, we have specific method for studying the protein composition of tissues, and we will take some example of this simple method. Means determining whether differences in protein composition exist in different tissues. Involves in investigating the expression of individual known proteins in such tissue. This method might involve the isolation of wildly differing amount of an individual protein, or none of the protein at all, or from different tissue. This means that we will isolate the proteins and see if that protein if this specific protein exists in that tissue or not. Also, it involves the detection of an enzymatic activity associated with the protein in the extract or only particular tissue. For example, we can see if the specific protein, for example, exists in brain or heart or kidney or liver or lung or muscle or spleen or testis or whatever, this specific protein exists or not. So any of these specific protein can be detected in different tissue if it exists or not. So if it exists, you can see clearly the band. If it's not exist, this protein does not exist, for example, a liver, so you can see the absence of the band. In this case, you can see the presence and absence, but also you can see the level of expression. For example, in the kidney, you can see this expression, the existence of that protein is a little bit much less than the heart, and the heart is much less than the brain. So this means that this protein exists much more in brain, and then a little bit less in heart, and then a little bit less in the kidney. And of course, we cannot find it here in the liver. So this means by that kind of methods, specific methods, you can determine if the protein exists or not exists, and also how much the quantity of that protein. One of these techniques like Western blotting. Western blotting, first protein are denaturated by treatment with detergent like SDS, and then electrophoresis in a polyacrylamide gel based on the size. After this separation based on the size, second Western blotting, the gel separated, the gel prepared proteins are transferred to a nitrocellulose filter, which is incubated with the antibody. The antibody reacts specifically with the protein present at a particular position on the filter. The antibody is then visualized by radioactive or enzymatic detection procedure. Intensity of the band observed will provide a measure of the amount of protein present in the tissue. So this is what is known as Western blotting, and you can see how this technique can be conducted. So isolation of the protein, and then do electrophoresis, normal electrophoresis, and then after that transferring the gel bands to a nitrocellulose membrane, and then after that hybridization with the probe. And of course, in the end, you can see something like this. If your band or protein exists, you can see single band. If not exist, you will not see anything like this. Of course, there is difference. Also, this can be visualized on the tissue, or this can be used in situ hybridization. Differences between different tissue in appearance and function are correlated with quantitative and qualitative differences in protein composition as assayed by antibodies to individual proteins. So we can do the same thing in situ, which means in the tissue. So we can see if the tissue have that kind of protein or not by something similar. Okay, the last two specific methods in situ hybridization or Western blotting is for studying specific protein. Now we'll talk about general method for studying protein composition. In that kind of methods, we have to extract all the protein from the tissue and see all the types of protein that exist or not exist in one single tissue, for example, as we can see, as well as examining the expression of individual protein in different tissues. It's also possible to use more general methods to compare the overall protein composition in different tissues. All the proteins present in the tissue can be subjected to one-dimensional gel electrophoresis and examined by using a stain which reacts with all protein. So investigate variation in overall protein content of different tissues. So you can see that there is different tissues, for example 1, 2, 3, 4, 5, there is different samples. Every tissue we extract the total protein content and then separate it by electrophoresis, and then after denaturation we can see what is the proteins that exist there and how many bands are there, and of course this can tell you a lot of information that can be used further for more investigations. But anyway, this is a general method, and of course this general method can be studied in the beginning, and then after that you need specific method to determine which band contain what kind of protein, and like this. So a more detailed investigation of protein composition of different tissue can be achieved by two-dimensional gel electrophoresis. In this case it's more specific a little bit, but it's also general. The protein are first separated on the basis of differences in their charge, and in a technique known as isoelectric focusing, and then a subsequent electrophoresis of the protein are separated by their size. Hence a protein moves to a position determined by size and charge. So in this case you can see that there is the first dimension, one dimension isoelectric focusing like this. So the proteins are separated, then after that the protein are separated vertically, so it's horizontally and vertically. This is second dimensional SDS page polyacrylamide gel electrophoresis. This technique called two-dimensional gel electrophoresis is also a general method, but it's a little bit more give a little bit more information. Thus, some spots of the protein are found in only or few tissues and not in many others, while others are found dramatically different abundance in different tissues. So you can see that here proteins present in a similar level, for example in both tissues, like that one and that one. So different similar levels in both tissues, like this one and this one are the same, this one is the same. Protein present in only one tissue, like that one here, so, present only in that tissue. Proteins present in both tissues but in different levels, like that one here, this one here, this one here, this one here. It exists in both but different levels of expression. So you can see clearly this two-dimensional this separation can give you a lot of information, but it's also a general method. Methods have been developed which allow the protein responsible for forming a spot of particular interest to be identified and characterized. The development of new field of study known as proteomics, in which the power of two-dimensional gels to separate a wide range of proteins is combined with the ability to study specific proteins individually. This is a field which we know as proteomics. Individual spots of interest are excised from the two-dimensional gel and digested into constituent peptides using a proteolytic enzyme like trypsin, for example. So by that way we can see what is the composition of that protein. And of course nowadays we have also other methods like internal sequencing and all that, but the individual peptides from a single protein spot are then analyzed by mass spectroscopy. This can involve both matrix assisted laser desorption, ionization, like it's called MALDI, which allow the molecular weight of peptide to be determined, and nano electro spray mass spectroscopy, which allow the amino acid sequences of particular peptide to be obtained. So this is something like sequencing in the DNA, and by this technique you can see what is the sequence of the polypeptide chain and how and what is the composition. These two techniques are performed sequentially in a tandem mass spectrometer, which first determine the molecular weight of a peptide produced by trypsin digestion and then fragment it further, allowing the sequence to be determined. It's something like this: the band excites and then individual peptides can be obtained, and then the digestion, and see what is the sequences of that protein. Of course nowadays we have more advanced techniques to study this. As trypsin always cleaves proteins after a lysine or arginine residue, it's possible to align the peptide sequence against that of a known protein and hence identify the protein responsible for original spot. This method can thus be used to identify an individual protein which shows an interesting pattern of expression in different tissues, or whose expression is altered in a specific disease. Tissue specific. Now we'll go to the second part, which is tissue specific separation of messenger RNA. And, there is also methods for tissue specific separation, to study tissue specific separation of messenger RNA, and also there is a general methods for that. So although it seems likely that differences in the messenger RNA population of different tissues do indeed underlie the observed differences in protein, it's possible that all tissues have the same messenger RNA species, and that production of different protein is controlled by regulating which of these are selected by the ribosome to be translated into a protein. As with the study of protein, both specific and general method techniques exist for studying the messenger RNA expressed in a given tissue. As we can see now, like Northern plotting, for example, this is a specific method to study if a specific messenger RNA exists or not. So we also have a probe for the messenger RNA, and then of course we isolate the total RNA, and then also electrophrases in the gel, and then have the labeled probe which for specific messenger RNA, and then we can see if that specific messenger RNA exists or not in the sample or not. So this technique called Northern plotting. So it's similar to the Western blotting, but instead of working with proteins in Western blotting, we will work here with messenger RNA or RNAs. In situ hybridization is also exists in situ hybridization, but in this case we will we'll work with in situ hybridization for the RNA, not for the protein. So to know if a specific RNA is expressed in a specific tissue or not, particular tissue or not. Methods studying expression in RNA isolated from particular tissue can be supplemented by methods allowing direct visualization of the RNA in a particular cell types. In such methods known as in situ hybridization, a radioactive probe specific for RNA to be detected is hybridized to a section of tissue of interest in which cellular morphology have been maintained. This is one of the figures that can give you how this technique looks like and what we are looking for in an example of that kind of technique. Analyzation, of course, of the position at which radioactive probe has bound not only allows assessment of whether the particular tissue is expressing the RNA of interest, but also of which individual cell types within the tissue are responsible for such expression. This technique has been used to show that the expression of some messenger RNAs is confined to only one cell. General methods for studying the mRNA. Methods for studying individual RNA can be supplemented, of course, by methods for studying RNA population. Early experiment in this area made by use of the root curve, and of course nowadays we have other techniques, but this was the early experiment. Complementary DNA or cDNA copy of the RNA is first made using radioactive precursors, so that the cDNA is made radioactively labeled. Then after that, you can see that after associating the cDNA and the RNA, the rate at which the radioactive cDNA reanneals or hybridizes to the RNA is followed. So first as we can see that we produce the RNA and then make the cDNA, and after making the cDNA of the RNA first using radioactive precursors, so that the cDNA is made radioactively labeled, and then after associating the cDNA and the RNA, the rate at which the radioactive cDNA reanneals or hybridizes to the RNA is followed. This is a scheme that can show you this technique and how it works. So mixed RNA populations synthesized labeled cDNA, and then after that melting and then reannealing, and then abundant hybridizes more rapidly. So when we see that abundant RNA is hybridized more rapidly, so this gives you a hint what kind of RNA exists more than others. And this is an example of typical root curve, and of course this is another root curve but in different tissues. So you can see that these kind of RNAs exist here more than here, and this is a typical figure that can compare between transcriptomics between different kind of tissues. Nowadays there is other general methods for studying RNA, which is a transcriptomic method, which is called now is a ship-based method. Ship's method or gene ships. In this process, more molecules of an abundant RNA will be present than of an RNA which is less abundant. The cDNA from an abundant RNA will find a partner more rapidly than one derived from a rare one. This is powerful method, thus allows the expression of profile of individual tissues and cell types to be obtained and compared in a manner which combines the ability of northern blotting or in situ hybridization to study individual messenger RNA, which is ability of root curve to look generally at the entire mRNA population. This means that this shape-based method like microRNA, microarray or RNA-seq are more powerful and give you more data, and of course nowadays is most widely used in all labs. So this is a figure that can tell you how this ship-based method looks like. In this method, so-called gene ships are prepared in which DNA sequence derived from thousands of different, of course, messenger RNAs are laid out in a regular array at a density of over 10,000 different DNA sequence per square centimeter. Of course, nowadays they have more and more developed ships for that kind of techniques, so the number could be much more. But anyway, this is the main idea is to have all the different messenger RNA are laid in a regular array at a specific density, and then different DNA sequence per square meter can be obtained. This is achieved either by spotting out cDNA clones derived from a different messenger RNA at higher density on a glass slide, or by synthesizing short oligonucleotides derived from each messenger RNA in situ in the gene chip. Like something similar to that kind of this figure. So something similar to that, so you can see how this techniques is developed. The use of this method also provide further support for the conclusion that the messenger RNA population of different tissues show both qualitative and quantitative differences. Conclusion of this lecture, you can see that the qualitative and quantitative variation in protein composition between different tissue is paralleled by a similar variation in the nature of the messenger RNA species present in different tissue. Also, we have to know that the abundance of the individual protein as determined by two-dimensional gel electrophoresis generally correlated very well with the abundance of the corresponding messenger RNA as determined by a gene chip approach. Hence, the production of different protein by different tissues is not regulated by selecting which of the common pool of RNA molecules is selected for translation by the ribosome. In order to understand these bases for such tissue-specific variation in protein and messenger RNA composition, is therefore necessary to move one stage further back and examine the nature of DNA encoding these mRNA in different tissue. Now we come to the practical, so we need the student to describe the overall differences between two methods for studying. Number one, tissue-specific separation of proteins. Number two, tissue-specific separation of RNA. And of course, it would be good if you also describe the difference between the general method and the tissue-specific and the specific method of proteins, and the general method and the specific method of RNA. Thank you for listening, and I hope to follow us on our YouTube channel and see more videos of the course, and subscribe and comment for further lectures. Thank you, and see you in other lecture soon.